completed. So this is about our own genetic material. You know anything about genetic material? Genetic materials are certain chemical molecules which are deciding the genetic features, the properties which are transferred from generations to generations. Where are these genetic materials present? They are present in almost every cell of the body. Almost every cell. Now, what is the nature of genetic material? Genetic materials in all organisms are actually nucleic acids, acidic materials in the nucleus. You have learned in the previous classes that the nucleic acids are deoxyribonucleic acids and ribonucleic acid. A human being or any other organism is made up of cells. How many types of cells are there in our body? We have so many types of cells. And in that cells, we have nucleus. In that nucleus, we have something called chromosomes. The chromosome is actually made up of deoxyribonucleic acid and also proteins. Some segments of the deoxyribonucleic acid are said to be genes. The genes are responsible for the production of proteins, and the proteins are carrying out the administrative and structural and catalytic and all other functions in the body. The proteins are playing very important roles in the body. So the body is controlled, all aspects of a living being is being controlled through the proteins by the nucleic acids. So we say nucleic acid is the ultimate genetic material. This is your syllabus, an introduction on nucleic acid and the composition of nucleic acid, the structure of purines and pyrimidines, nucleosides, nucleotides. You know that nucleotides and nucleosides, etc., are the component structures of nucleic acid. ATP is a nucleic acid having a variety of functions. The most important role is its function as the energy currency of the cell. And you have cyclic adenosine monophosphate, CAMP, cyclic adenosine monophosphate. It is actually a part of the messenger system in the body. It is actually said to be a second messenger in the body. And you have the DNA and the RNA, which are the genetic materials, or we say which are the nucleic acids, and their structure, their types, and the Watson and Crick model of DNA. You have heard about Watson and Crick. You see, nucleic acids are actually made up of individual units called nucleotides. Several 100,000 nucleotides join together to form nucleic acids. A nucleotide in turn is made up of a nucleoside and a phosphate. One nucleotide consists of one nucleoside and one phosphate. Sometimes two phosphates, sometimes three phosphate. Accordingly, we call it monophosphate, diphosphate, triphosphate, etc. You know, adenosine triphosphate, ATP is a triphosphate. Adenosine monophosphate, AMP, that's a monophosphate. ADP, adenosine diphosphate, consists of two phosphates, like that. And a nucleoside in turn is made up of a nitrogenous base and a pentose sugar. What's a nitrogenous base? A base material, a ring structure, a heterocyclic compound containing nitrogen. What's a pentose sugar? You have learned earlier that the pentose sugars are actually 5-carbon sugars. It is deoxyribose sugar in DNA and ribose sugar in RNA. So let us come to the structure of these components in detail. Ribose. How many members are there in the ring, including oxygen? 1, 2, 3, 4, 5. This is a 5-member ring, heterocyclic in nature, and this is actually ribose sugar. Why is it a pentose? You see numbered carbon atoms. There are 5 carbons, so this is a pentose sugar. Look at this red colored OH group on carbon number 2. Make a note of it because this is the structure of deoxyribose. What's the difference between deoxyribose and ribose? A more proper name is actually 2-deoxyribose. Why 2-deoxyribose? Because deoxy, oxygen is removed from carbon number 2. Look here, on carbon number 2 of deoxyribose, oxygen is not there, which was present actually in the case of ribose. So the difference between ribose sugar and 2-deoxyribose sugar is the presence or absence of an oxygen atom on carbon number 2. And really that makes a lot of difference because when they are forming a nucleic acid, this OH group on carbon number 2 will be the only free OH group. All the other hydroxy groups are reacted with the other components of the we say nucleic acid. So what is going to be free is this OH group, and you know that OH group is polar in nature, it is hydrophilic in nature, it is highly reactive in nature, so it makes the RNA molecule less stable and more reactive. While in the case of DNA, such an OH group is not there. Therefore, this deoxyribose containing molecule, the DNA is less reactive and more stable. That's why, my dear children, you know, RNA viruses are prone to mutations. RNA is undergoing reactions and RNA is highly mutative in nature. I mean, it can undergo mutations very fast. Of course, you know that in the recent coronavirus incident, new strains are appearing in different parts of the world. The major reason is that coronavirus is actually an RNA virus and not a DNA virus. Why DNA is more suitable to be the genetic material? You have studied in 12th standard that the genetic material should be more stable. It should not undergo changes according to the age or sex of the organism. And DNA is more suitable because of the absence of an OH group on carbon number 2. That is just one of the reasons. So of course, some other reasons are also there. We will see it in the future. And we were talking about nitrogenous bases. We studied earlier, sugars plus nitrogenous bases is equal to nucleoside. So what about the nitrogenous bases? These bases are planar, aromatic in nature, they are heterocyclic in nature, and they are derivatives of purines or pyrimidines. What's a purine? This is a typical purine, a general structure for a purine. Of course, side groups may be there when we come to the case of nitrogenous bases of the nucleic acids. You see, how many rings are there? Ring 1, ring 2. So purines are double ringed compounds. And you see, on the ring there are nitrogen atoms. Remember, this nitrogen is present as a part of the ring. Nitrogens are present as a part of the ring. Nitrogens 1, 3, 7 and 9. And remember, this double bond is always present in all the purines. This double bond is present. Make a note of it. Sometimes children may draw a structure like this. They may draw a figure. They draw a figure like this. Okay, up to this much is okay. Sometimes they draw this nitrogen like this, nitrogen number 7. It is wrong. Don't do this. They may do they may draw this nitrogen like this. That also is wrong. Why? Because this nitrogen is supposed to be a part of the ring. Within the ring, this also is a part of the ring. The same is applicable to this nitrogen also. So if you draw the nitrogen atoms outside the ring, if I am evaluating the paper, your mark will be 0. Got it? Okay. And the pyrimidines, how many rings are there in the pyrimidines? Only a single ring. How many nitrogen atoms are there? 2. Both are within the ring. Of course, additive side groups may be there. Don't worry about it. In the basic pyrimidine structure, there are 2 nitrogen atoms as a part of the ring. Okay? Be careful to draw them within the ring, not outside the ring. And these are the different types of nitrogenous bases. This is a basic structure of pyrimidine. Look at the number. We begin from here, the numbering, and we reach here. These are the 3 types of pyrimidines in the nucleic acids: uracil, thymine, cytosine. You should draw their structure. It will come for the examination. You see, on this nitrogen, always there is a hydrogen atom. Always there is a hydrogen atom. While on carbon number 2, always there is a double bonded oxygen. And or actually the third member, it is a nitrogen atom. There is a hydrogen atom in both the uracil and thymine, while it is not there in cytosine. Because on the fourth site, there is a double bonded oxygen in both uracil and thymine, while it is an amino group in the case of cytosine. And what's the difference between uracil and thymine? You see, on carbon number 5, a methyl group is attached in the case of thymine, while no such side group is there in the case of uracil. Actually, we should say thymine is the cousin of uracil. They are related in structure. You have studied in lower classes, in your higher secondary classes, that RNA was the first formed nucleic acid. Then by editing the RNA, DNA molecule is produced. Why the DNA was required? Because DNA is more stable. Why the DNA is more stable? The second reason is this one. It contains thymine instead of uracil. See here, uracil is present only in RNA, while thymine is present only in DNA. What's the importance of thymine? This methyl group is more hydrophobic in nature. Methyl group is more hydrophobic in nature. It will make the DNA more stable. Okay, so make a note of it. The difference between uracil and thymine. So that's why DNA is more stable. Of course, cytosine is present in both RNA and DNA. Coming to the pyrimidines, coming to the purines. Look at the numbering pattern of the purines. It is like this and like this. This is adenine. In adenine, there is an amino group on position 6. While in guanine, at position 6, there is a double bond oxygen. While at position 2, there is an amino group. That's how they differ from each other. And you should learn how to draw this figure to come for the examination. And we need to learn something in more detail. You have learned earlier that adenine is hydrogen bonding with the thymine. Why? Because of the position of the hydrogen donor and hydrogen acceptor. We will come to these topics in other slides. And how do they join together to form the nucleotides? Look here, a ribose sugar is joining with a nitrogenous base. This is actually a nucleoside. The base is joined with carbon number 1 of the ribose sugar. Which base or which atom of the base? You see here, you can see this NH. This NH is present in all purines and all pyrimidines because it is going to join with carbon number 1 of the pentose sugar. So the nitrogen of the nitrogenous base is joined with the OH group of the Pender sugar. Remember, for the Pender sugar, this OH group on carbon number 1 is upwards, which means it is a beta sugar, beta D ribose molecules or beta D deoxyribose molecules. Okay. Now this nucleoside joins with the phosphate groups, making it nucleotide. So this is a ribonucleotide because this OH is present, while here OH is absent. Therefore, this is a deoxyribonucleotide. Draw this structure several times. You need to draw it for the examination. This is the structure of ADP, adenosine diphosphate. What is this? This is the ribose sugar. Ribose sugar. Here you have the CH3, that is CH2 group, that's actually not mentioned here because this bend is indicating carbon. This is a nitrogenous base. What do we call it? Adenine. Look at the arrangement of the adenine. The nitrogen is bonded with the OH group, and this is a beta ribose. And on carbon number 5, there was the CH2OH. That OH is reacting with the phosphoric acid, and this is now a diphosphate because phosphate number 1, phosphate number 2, this is a diphosphate. So adenine plus sugar is adenosine, adenosine diphosphate. And one more point, remember this OH group is ionizable, which means this hydrogen may leave as a proton and this oxygen may get a negative charge. The same is applicable here also. This proton may leave here and this oxygen may get a negative charge. Why do we say phosphoric acid is an acid? An acid is a donor of protons. So here this is a phosphoric acid, donor of protons. So it is donating protons. Here this is a diphosphate. So totally 2 negative charges are there. By the relieving of the protons, these oxygens are assuming negative charges. Look here children, nucleotides are negatively charged, and several 100,000 nucleotides join together to form a nucleic acid. The nucleic acid also is negatively charged because of the negative charge on the phosphate group. Make a note of it. The same is applicable for this OH also. That also may get a negative charge. Coming to ATP, adenine plus sugar is adenosine plus how many phosphates are there? Phosphate 1, phosphate 2 and phosphate 3, adenosine triphosphate. How many negative charges are possible? Actually in the physiological conditions, these molecules are ionized. So how many negative charges are possible? 1, 2, 3 and 4 negative charges are possible, and this is the adenosine triphosphate molecule, the ATP, the energy currency of the cell. Remember, on the ATP, ribose sugar is present and not deoxyribose sugar. And AMB is adenine plus sugar, adenosine plus 1 phosphate, adenosine monophosphate is AMB. While this monophosphate can become a cyclic molecule. How? This is carbon number 1, 2, 3, 4 and carbon number 5. It is bearing an OH group, or on the OH group there is a phosphate, on the phosphate there is this OH group. This OH group may react with this OH group on carbon number 3. So forming a new bond. Actually this is an ester bond between acid and alcohol. This is an ester bond. Now this is a cyclic molecule. This is cyclic adenosine monophosphate. Remember, you will study this molecule in your 4th semester. And nomenclature of different sugars and their derivatives, etc. we will see in the next slide.